So for today's lab activity, we are going to be able to basically perform the central dogma of biology. And so you guys already know this. And so what is a simplified version of the central dogma of biology? We are taking DNA and we're going to use it as directions to build RNA. Now that RNA has got to be processed before it's in the correct form, known as messenger RNA, mRNA. And then we can take that mRNA and use it as directions to be able to make proteins. So DNA is used as directions to make RNA. RNA is used as directions to build proteins. So the process of using DNA in order to make RNA is known as transcription. And then the machine that is responsible for doing transcription is known as RNA polymerase. So RNA polymerase is going to make our RNA. Now our RNA in our eukaryotic organism is not going to be ready to leave the nucleus until it's been processed. And so processing means it needs a 5' cap, it needs a 3' poly A tail. But what we're really going to focus on in this particular lab activity is it needs to undergo splicing. And so what is splicing? Well, splicing is where we remove introns in order to connect exons, because exons are the coding part of the RNA. So once you have that final version of the RNA called the messenger RNA, then we can go and use that as directions to build proteins, and that process is known as translation. And the machine that's responsible for translation is known as the ribosome. So what we see depicted in the middle is a human genome sequence, and it is about 600 nucleotides long. And what we're going to ask you to do in this particular lab activity is we're going to ask you to do transcription. We're going to ask you to do the splicing component of RNA processing, and then we're going to ask you to do translation and build us some protein. So what I need for you to do is I need you to go to your lab manual, the hard copy of your lab manual, and I need you to find this particular page. And so if you have the spring 2020 version, that's page 147. So get that page out because we are going to draw all over it. So let's begin. We want to focus on the process of transcription, okay? And so it's like, well, if you want to do transcription and you're looking at this DNA sequence, how does RNA polymerase know where in the DNA sequence to grab on, to bind to? So that's really going to be the question of where does RNA polymerase grab onto the DNA? And so what we'll have to remember from lecture is that it's looking for a particular pattern of nucleotides called the promoter sequence. So again, this is a specific pattern of nucleotides. And it's like, well, what is the pattern? We go 5' T, A, T, A, T, T, A. So that would be the top strand, the 5' to 3' strand. And then the bottom strand of DNA would be complementary. So if I have a T, I put an A. If I have an A, I put a T. And then you just, solve for the rest. And so we're looking for this particular pattern within the DNA. Now, what's nice about this particular image that you're looking at is that we can number the nucleotides. So we start with 1 and we end with 600. In this particular slide, what we're seeing is I'm showing you 300 of the nucleotides. The numbers that are depicted here on the right-hand side, these represent the number of the very last nucleotide in that strand. So this A and this T represent position 60 in the 5' strand and in the 3' strand. This G and this C in the second row represents position 120 in the 5' strand and in the 3' strand. this A and this T, they both represent position 180. One is in the 5' to 3' strand, and the other one is in the opposite strand. Same thing with the remaining row. So we can figure out the very last nucleotide of each row, what number that is. We can also figure out the very first nucleotide. And so if we ended on 60, in the first row, this G and this C, oh, that's position number 1. Okay, we go to the second row. If the first row ended in 60, the second row has to begin with 61. If the second row ends with 120, the third row has to begin with 121. If the third row ends with 180, the fourth row, those nucleotides that begin it have to be 181. And then the fifth row, if we ended with 240 in the fourth, the fifth has to begin with 241. Right? So we've got the edges all labeled out, which is super useful. But I also like to kind of have a reference point in the middle of my sequences to make things a little bit easier. And so you want to get in the habit of it. It just makes it so much easier. So if we go to the second row and we go to this particular T where I'm putting the arrow, that T is going to represent nucleotide 90. If we go to the third row, the T that I'm labeling in the third row is going to represent nucleotide 150. If we go to the fourth row, the C that I'm labeling in the fourth row is going to represent nucleotide 210. And then if we go to the fifth row, the T that I'm indicating is going to represent nucleotide 270. So now we've got both ends labeled and we've got the middle labeled. So what I want you to do is I want you to push pause and I want you to label your own because again, it's really going to be important that you do this. Okay, so I'm assuming that you did this, great. Now, what I'm going to ask you to do is I'm going to ask you to take on the role of RNA polymerase. And so the role of RNA polymerase is I need to look at this DNA and find the pattern TA, TAA, TTA. So I want you to push pause and then on your piece of paper, I want you to scan along the top strand on each row and try to find TA, TAA, TTA. Go ahead and push pause now. Okay, are you back? So again, we want TA, TAA, TTA. Hopefully you were able to find it. So on the second row, right here, TA, TAA, TTA, that's the 5' to 3'. And if we're going 3' to 5', AT, ATT, AAT. So where I've highlighted in yellow, this represents the promoter sequence. So here, this is the promoter. This is where RNA polymerase is going to grab onto the DNA. It's like, okay, I got a hold of you. Now, how do I know where to begin initiating transcription? And so what's interesting is that there's patterns to it. And so what they've determined is that once you find the promoter region, you can determine where transcription can start. So here, I'm kind of outlining that promoter region. And it's like, okay, what nucleotides are the beginning and the end of the promoter? So notice how I have that 90 nice and labeled. So we just count backwards from that 90, right? So 90, 89, 88, 87, 86. Oh, this A is nucleotide 85. Okay, that's good. Now we can keep counting back to the very first T of the promoter, and that's going to be position 78. So this T is position 78. So the promoter is a pattern of nucleotides in this particular sequence that's between position 78 to 85. If I want to know where to start transcription, I have to go 25 nucleotides downstream of the promoter in the 3 to 5 strand. And what I'm looking for is I'm looking for a T at that position. Now, you could literally go count, I'm not a big fan of counting if I can narrow down how much counting I have to do. So it's like, well, you said it's 25 nucleotides downstream. So if we, at the very last nucleotide of the promoter is 85, if I were to take 85 and add 25 to it, that would mean it's position 110. And you told me that I need to be looking at the bottom strand, the 3' to 5' strand that you're putting an asterisk on the left-hand side. So, okay, what I'm going to do is I know that that C, the very last C of that strand is 120. I'm just going to do a quick and dirty count backwards. So I got 20, 19, 18, 17, 16, 15, 14, 13, 12, 11. I found you. I found that T at 110. And so what I'm going to do is I'm going to highlight that T for you. So there's that T. And so that T is found at position 110. And this is where we initiate transcription. This is where RNA polymerase goes, okay, I can begin building the RNA. Fantastic. Okay, cool. So promoter told us where to grab onto the DNA. The T, 25 nucleotides downstream of the promoter, told us where RNA polymerase is going to begin building the RNA. And then we're going to have a place that tells us where to stop. Okay, so that's going to be your terminator sequence. And so if we go to the next slide, what I've done is I've zoomed in on sections 300 to 540 of your particular lab report. And so again, with this, what I recommend you do is you label the ends. So we know that, the last nucleotides here are 300. We know that 360 on the second row, 420 on the third row, 480 on the fourth row, and 540 on the fifth row. So what's the position here? Well, with regard to this one, what we have to remember is the position before it. So this one is going to be on the left-hand side. This would be 241. Second row, the beginning is going to be 301. Third row, it's going to be 361. Fourth row, it's going to be 421. And fifth row, it's going to be 481. Okay? And so we could go through here and label some in addition to like what we're seeing above. So I could go to this particular T in the first row, and that would be position like 270. I could go to the third one, and this G happens to be position like 330. I could go down here to this T right here, and that's going to be position 390. And then I'm going to get you to put in, the rest of them. So put in the rest of them, do a little counting, put in the rest of them, and then once you're done, let me know, so to speak, and that you're ready to go. Okay, so we're still doing the process of transcription. And so we already found the promoter, we found the initiation site for transcription. So now what we need to do is to end transcription, we need to be told where to end. So we need a terminator site. So it's like, okay, a terminator is going to have to be a specific pattern of nucleotides. And so what we're going to do is we're going to look at the 3' to 5' strands, so the bottom strand of each row, and we're looking for the pattern 3' TTA TTT. So 3 T's in a row. Boom, boom, boom. Now I want you to put it on pause. I want you to scroll through and look for that particular pattern TTA TTT in the bottom strand, the 3' to 5' strand. So go ahead and push pause and see if you can find it on your own. Okay, so were you able to find it? Did you find it? So again, we're looking for TTA and then a triple T. So if we look at this strand right here at the bottom, TTA, triple T, there is our terminator in our fifth row, the 481 to 540 row. And so we would label this particular one the terminator sequence. Okay, so you've basically just done transcription by being RNA polymerase, and that you found the major components. Now, if we wanted to full-on do transcription, what we could do is in blue, I could show you what it looks like. So if we started on the top row, you are not required to do this. Okay, this is just showing you if you were actually RNA polymerase. If I saw a T, I would put an A. If I saw an A, I put a U. If I saw an A, I put a U. If I saw an A, I put a U. And then you just keep building until you hit the terminator sequence, and then you'd stop. And so that would be transcription. And so now we have this RNA that we can work with, and we would put a 5' cap on it, we would put a poly A tail on it, but the part that you're doing in the lab activity is that we need for you to be doing the splicing. So you're going to be acting like the spliceosome, and you're going to be able to identify the 2 introns that are found in this sequence. So if we go back to the beginning of your genome sequence, where we started our initiation, right? So remember transcriptional initiation, which was 25 nucleotides downstream of the promoter, so at position 110. Between that and the very last nucleotide 300 shown in this particular image, we need to find the intron. There's 1 intron located in between this. And so it's like, well, how do you figure out where the intron is? So the intron has a beginning, a branching point, which is the middle, and an end. And so what we want to do is when we want to be doing this process of splicing, we need to find the intron component. So we need to find the branch component. And so the branch component is a pattern of nucleotides, and it's going to be our 5' to 3' sequence. And I need for you to find, if it was RNA, which was where this would be happening, it would be UAC, UAAC. Now, because we're dealing with DNA, when you're looking at this actual sequence right now, it would be TAC, TAAC. So what I want you to do is I want you to put it on pause, and I want you to scan between position 110 and position 300 in the top strand and see if you can find TAC, TAAC. So go ahead and push stop and see if you can find that particular pattern of nucleotides. Okay, hopefully you found it. We got goals. So I'm going to show you where it is. If we look at the 5th row, we have TAC, TAAC. So right here that we're looking at, okay? And so that represents our branch point. So from here to here, that's your branch. So this is found within the middle of the intron. Now, we want to be able to find the beginning of the intron and the end of the intron. It's like, well, how do you do that? So to be able to find the beginning, what we have to do is we have to take the beginning of the branch. So you take the beginning of the branch nucleotide number, and I need you to subtract a designated amount. And in this particular case, I need you to subtract 71. So we're going backwards 71 nucleotides. So I need you to know what's the nucleotide number of the branch. And so in this particular case, the branch, the beginning is position 255, and the last position is 261. So if I want to figure out where to find the branch beginning, or sorry, the intron beginning, because I'm looking for the GT near that position, what's going to happen is I'm going to take 255 and I'm just going to subtract 71 from it, and that will get me near where I need to be. And so that puts me at position 184. So what I expect is at positions 182 and 183, I should find the G and T. So, okay, here's 181 in the 4th row. Oh, 82, 83, look, there you are, GT, because here's that 184. So position 182 is the beginning of the intron. So this is intron number 1. So it's like, okay, Dr. Strong, you found the beginning, but where is the end? Well, to find the end, so if we want to find the end of the intron, what we need to do is we need to take the last branch nucleotide, and I need to add 25 to it. And then once I get to that, plus 25, the pattern that I'm looking for is AG. So I told you that 261 was the last nucleotide of the branch. I need to add 25 nucleotides to that. So 261 plus 25 is 286. I need to go find position 286 because 287 and 288 should be an A and a G. So, okay, here's 270 in the middle. And I said I need to go find 286. So 70, 1, 2, 3, 4, 5, 6, 7, 8, 9. Here's 280. 1, 2, 3, 4, 5, 6. There's 286. That's C. And look, there's my A and G. So 288 is the end of the intron. So basically, when you go to do the process of translation, you need to ignore nucleotides 182 to 288. You need to cross them off. They don't exist for you anymore. They are gone. You throw them away. And so one way you could do that is kind of highlight them. You start with that G and it's gone. You just see how I'm crossing this off? You're gone. And I keep going all the way till I get to that G. Now, nucleotide 181, you count. Nucleotide 289, you count. I have to use you in the process of translation, but everything else you got crossed off. Now, I'm going to start you off a little bit with translation, but I also have to show you where the second intron is. So we're going to start translation, but we're not going to finish it in this video. You're going to have to finish it on your own. So when we want to start the process of translation, so to start translation, what do I have to find? And you should remember this from lecture. I need to find a start codon. And do you remember what the start codon pattern was? Because it's pattern of 3. So the start codon is AUG, right? And so if we're looking at the DNA, it's going to be ATG. So I have to look at where did transcription begin, and I'll look past that up until the first intron. So somewhere in there, there's going to be an ATG, and I just got to find that ATG. And it's going to be in the 5' to 3' strand. So it's like, okay, starting at position 110, 5' to 3' strand, top strand, do I find an ATG? I scan, I scan, I scan along. Do I see it? Do I see it? Do I see it? And then I go to the next strand. Okay, I'm looking for that ATG. I'm looking for that ATG. And then I find it. There you are. There's that ATG where I'm highlighting in green. And so that ATG is going to be in the 3rd row. And it's going to be located just at near the end of the 3rd row. And so if we count backwards, the A of the 3rd row, that's 180. So we go 80, 79, 78, 77. So the ATG starts at 176. So this is where you begin translation. So we read patterns of 3. And what you should do is you should write down the patterns of 3 so that you can translate them later using the codon table. So we've got ATG, first codon. Then we go GA, and then we go to the 4th line, A. GAA, second codon. Then we have to skip the first intron, right? We can't use any of those nucleotides. And so my 3rd codon begins at TTG. And then I keep going, zoom, 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 until I get to the second intron. So if we want to identify the second intron, what are we going to have to do? Is we're going to have to look for particular sequence, right? And so again, we are trying to do this splicing. We're trying to identify the introns. And so we're trying to identify intron number 2. And so the particular pattern that we're looking for intron number 2, if it were RNA, it would be UAC, UAA. So remember, because we're showing you the DNA, it would be TAC, TAA. So what I want you to do is I want you to scan through, see if you can find where is this, and this should already be nice and numbered up for you from previously, right? So scan through in the 5' to 3', find TAC, TAA. Make sure you find it. Have you found it yet? And I'm kind of drawing this out because I don't want you just to rely on me automatically doing it. So maybe if I pause a little bit longer, you're more likely to want to do it yourself. Hopefully would be the idea. Not guaranteed, but you know, we got goals. All right, I'll highlight it for you. So we got TAC, whoops, kind of messed those. TAC, TAAC. So where I've highlighted, that is going to be your branch sequence of your intron. So this is your branch. So what is the beginning of your branch? That nucleotide T at the beginning is position 391. That particular, oops, that one went a little too far, didn't it? TAC, TAA. It got those two. There you go. The A right there, right? Yeah, the A right there is going to be, we just count forward, so 91, 92, 93, 94, 95, 96, right? So 91, 2, 3, 4, 5, 6. So you got 396 right there. And then we just go from there, right, guys? So we got TAC, TAA. Oh, I think that actually we should have added a C at the end of that. I'm pretty sure actually there was supposed to be a C at the end of the branch. So that was my little error. Let me highlight it. I was right to begin with. So highlight that C. And so in actuality, our branch is going to end with 397. So that was my mistake. So our branch is 391 to 397. If we want to know where the beginning is, so where is the beginning of the intron? What we need to do is we need to take the beginning of the branch, which is position 391, and we need to go and we need to subtract 53 nucleotides from it. And so that's going to give us position 338, and what I expect to see is that 336 and 337, I better find a GT. So I should see the letters GT at 336 and 337. So it's like, okay, go find 338. And so we just, you know, we got 360, you got 301 is right here, right? We've got 361 is right there. So we kind of know where we need to start looking with regard to the middle. So you know it's going to have to be in the middle of the 360, the second row. And so what we could do is we could, you know, literally count backwards or forwards. It's up to you how you want to do it. So we'd be like, this is 60 right here. 60, 59, 58, 57, 56, 55, 54. We'd count, we'd count, we'd count, we'd count. It'd be just tons of fun, right? And so what we'd end up finding is that the GT that we're after ends up being at position 336. So it's going to be right here. Whoops, can't use the dark purple. It hides it too well. So we'll go yellow. So this one right here. So it's going to be at 336 is going to be the beginning of our intron. So the start of intron is 336. Okay, so if we want to figure out where the end of the intron is, so where is the end of the intron, we need to take the ending branch nucleotide, and in this case the ending branch nucleotide number was 397, and I need to subtract from it, or sorry, add to it. In this case, when we're looking for the end, we add to it 25. So 397 plus 25, we end up getting 422. And so at position 423 and 424, I'm looking for the nucleotides AG. So we just look, oh, row number 3, that's 420. There's 421. There's the C right here, 422. There you are, 423, 424. So the end of the second intron is going to be position 424. So what we would need to do is we would need to throw away what's found from the beginning of the intron. So from here, zoom, 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 just get rid of it. You're not going to be using it in the process of translation. So we would use the sequence before it, and we use the sequence after it, and we would continue the process of translation until we were told to stop. And so how do we know when to stop? So with translation, the way that you know to stop is you hit a stop codon. So you're going to keep translating these components in your big picture until you hit a stop codon. So follow the remaining directions in your particular handout. Here is a codon table that gives you the single letter codes for each amino acid. So here's the amino acid name where I'm highlighting phenylalanine. Here is the single letter code for F. Leucine, it's going to be L. And then you just go through and you'll see that there's going to actually be a message written in this code. And then you should be good to go from there. And then you turn in your lab report, answer all the questions, do what you need to do. All right, that's the end of this lab activity.